Joining us today in person and on Zoom for Neurometabolism 101. This is part of the Harvard Brain Science Initiative's 101 series. So today it's my pleasure to introduce Professor Gary Yellen, who is a professor here in the neurobiology department at HMS. And his lab is interested in the interplay between neuronal metabolism and excitability, and why it is that metabolic changes can have such dramatic effects on brain excitability. And in particular, one key example that drives some of his lab's research is the ketogenic diet therapy, which is a diet therapy which can be effective for epilepsy that does not respond to standard pharmacological treatment. The lab uses a mix of electrophysiology, pharmacological tools, mass spec imaging, transgenic mice. In particular, one of the exciting things they do is designing and using fluorescent biosensors that are genetically encoded and can detect and visualize really rapid metabolic changes. Gary did his undergrad here at Harvard, his PhD at Yale, postdoc at Brandeis, and he started a lab at Johns Hopkins. And initially, the lab was studying the moving parts of ion channels, how it is that they open and close. So I wanted to thank you so much, Gary, for joining us. And just a couple housekeeping notes. If you're joining on Zoom, feel free to type messages in the chat at any time. Or at the end, you can also raise your hands to ask questions on audio. And if you're joining us for lunch, just hang out here at the end of the talk.
Well, thanks, Perzad. Oh, I hope it's not too loud. I guess I have to whisper into it. Thanks all for coming. I haven't actually taught a course on neurometabolism, so this was an interesting exercise, and we'll see how it goes. So before I get into the substance of the talk, I just thought I'd address the elephant in the room. Or maybe it's an elephant that many of you gotten past because you're actually in the room or watching on Zoom, which is why should you even care about brain energy metabolism? So first of all, I'm going to focus on energy metabolism. Metabolism refers to basically any biochemical transformation, but I'm going to focus on energy metabolism. So why should you care about brain energy metabolism? Isn't it just one of those things that takes care of itself, and we can worry about the interesting stuff? And there's a couple of reasons, and I will get into more details in the talk itself. Brain metabolism is very intense. I'll say what I mean by that in a few minutes, but it's a clue that it's important. It is highly dynamic, both in time and in space. So it varies, between different parts of the brain when they're active. It varies in seconds, in a part of the brain that's active or in neurons that are active. It is absolutely crucial. You can't do without it for more than, let's say, tens of seconds, depending on how you measure it. And you could get permanent brain damage in 2 minutes. So it's extremely crucial. And if it's disrupted in disease, either by metabolic disease or perhaps in other kinds of diseases that we don't think of as being primarily metabolic, then it can have pretty severe consequences. And finally, and this is the reason I think people are a little prejudiced against thinking about brain metabolism, is it was called housekeeping for a long time. You get the metabolic pathways chart on the wall and say, well, all of that is housekeeping. Now we can study the interesting stuff, like how the brain thinks, or what switches on in a cancer cell to make it a cancer cell. And it was really the cancer metabolism people that figured out, well, this is probably a mistake to dismiss it as housekeeping. The cancer metabolism people realized that, after spending decades cloning oncogenes, viral oncogenes, cellular oncogenes, they came to the realization that pretty much all these oncogenes regulate metabolism, and that metabolism is really a central player in cell fate, both in cancer, a really dramatic case, but also in development. And there are people in the Harvard area working on how important neuroenergy metabolism is to development. So these are the reasons. I put in a note that housekeeping was viewed as was kind of putting metabolism in its place for a while. But although my grandfathers may have thought of housekeeping as something that took care of itself, my grandmothers didn't, and probably none of you do. So, and you know how bad things go if you don't attend to the housekeeping for too long. So anyway, so now I'll switch to what's the plan of the talk. So I'm gonna cover, it's actually a very big area, and I'm gonna cover five littler topics. And so the basic scheme of the talk is I'm going to start with a top-down thinking about the system. What are the constraints on the system? How does the whole system behave? What does the whole brain do for energy? And then switch to a bottom up. Like, what does this look like at the cellular level? What do energy demand and supply look like at the cellular level? Then I'll switch to a topic I advertised already, which is dynamics. I put this as what happens to brain energy metabolism when you think hard. But what I'm really going to talk about is when a particular part of your brain thinks hard. Although actually, my son has told me that championship chess players actually burn an enormous number of calories during a match. And I don't know how much of that is anxiety, but but it may actually be that their supercomputers are working extra hard during that time. But I'm not going to talk about that. I'm going to talk about what happens in particular brain areas when they think hard. Then I'm going to switch again. This is a sort of top down and bottom up again. Is that all energy is local, or all energy is really cellular. We can think about the energy in the whole organ, but the energy has to be generated inside each cell. And to think about how, when energy use is high, how cells might relate to each other in handling that energy demand. And then finally, Parizad referred to this before, I'm going to talk about fuel choice and glucose and the alternative fuels to glucose. And Parizad mentioned that this is really how I got interested in this area, is the ketogenic diet as a treatment for epilepsy. And we think that fuel choice, basically which fuel a cell is using to produce energy, may have very interesting and important functional consequences. So that's the basic plan of the talk. As I said, I've never given exactly this talk before, so we'll see how it goes, but feel free to interrupt. I guess the people on Zoom will type messages to Parizad, but you guys can just blurt stuff out, but we'll jump right in. So the brain, the top-down analysis is the brain is a remarkably energy-intensive organ. So, just comparing your brain to the whole body, these are sort of rough average weights for an adult male, is that the brain might weigh 1.4 kilograms, the body 70 kilograms. That's for a nice fit adult male. And that's 2% of your brain, of your body mass is your brain. But oxygen use, you can measure oxygen use. I'm not going to talk in detail about how all these things are measured, but it's an important and interesting subject. I'm happy to discuss it if you want. But oxygen use, you can imagine doing blood gases to measure, how much, and blood flow to see how much oxygen is extracted by the brain. And the calculation is that your brain uses about 50 mils per minute of oxygen. Air is 20% oxygen, so that's 250 mils of air is what you need to breathe to just fuel your brain, compared to 250 mils of oxygen for your whole body. So 20%, which is, so the brain is obviously batting way above its weight in terms of energy use. And this had to be an important evolutionary decision, right? This is a big deal. You don't waste 20% of your body's energy unnecessarily, because as organisms, we need to go out and forage and find food to supply our energy when times are tough. When times are tough, you can't cut back on that very much. You actually can't cut back on that 20%, on that 50 mils per minute in your brain. Just to put these numbers in perspective a little bit, so just to re-emphasize, the brain consumes about 20% of the body's total energy budget. That's a big deal. And if you put this in terms of power use, The brain is, so your whole body is about 100 watts. The architects who designed this room took into account how many people could sit in it and multiplied by 100 watts to figure out how much extra cooling they would need when the room is occupied. And a fifth of that is your brain. So your brain is like a 20-watt. I mean, most of us, most of us still understand what a 100-watt light bulb does, right? These are like 100-watt, 200-watt equivalent light bulbs that take almost no energy. But a 100-watt light bulb, you don't want to have too many of those burning in your house at once. It's substantial energy. You could think of this as also a very cheap supercomputer, right? If you think about what the power dissipation from supercomputers, if we could do all blockchain with brains, then the world would be a less energy intensive place. Now, again, to just some more numbers to put this in perspective. How does that power use correspond to a turnover of ATP? And the rough calculation is 5.5 millimolar per minute. So that means that in 1 minute, you could burn that much ATP. Now, of course, again, putting this in perspective, cellular ATP is on the order of 3 millimolar. So that means you would burn all your ATP in a cell if you could do that. Because you don't, it's not worth anything. After you burn a certain amount of ATP, the ADP accumulates, it's just not worth anything anymore. You don't get as much energy out of it. So you have to be constantly resynthesizing ATP to keep up with this. And that's what we're going to talk about when we go to the cellular level. And luckily, actually, people don't think about this too much in the brain. Everybody knows that muscle has a lot of phosphocreatine, right? Phosphocreatine is a high-energy, so-called high-energy phosphate compound. It can donate its phosphate to an ADP to make it back into an ATP. And the brain cells, neurons, also have this energy reserve. It's a bit lower in muscle. It's like 20 or 30 millimolar. In brain cells, it's estimated to be more like 10 millimolar, but still pretty substantial. So that's a good backup. That's the first backup to the ATP reserve. And so that's a good backup to know about. In terms of backups, though, the brain doesn't have a lot. Local reserves of energy are quite limited. First of all, oxygen, which you need to produce a lot of the energy in the brain, there's like no store. The only store of oxygen in your brain is like if you stopped your circulation entirely would be the oxygenated hemoglobin that remains in your vasculature. And that's not much, right? That will go away really quickly. But in terms of carbon sources for energy, the only real local reserve is glycogen. Glycogen, it's there at about an equivalent of 6 millimolar ATP. And it's mostly in astrocytes, so some is in neurons. It's still a little mysterious exactly when and how the glycogen is used. But to compare this to glucose use, we burn when we use that much oxygen, we burn about 0.3 millimolar glucose per minute. Now, circulating glucose is on the order of 5 millimolar. And but in the extracellular spaces of the brain, it can be down at around 1 or 2 millimolar. And we burn 0.3 millimolar per minute. So that looks like it's 20 minutes, but that's probably an overestimate because if you have an ischemia, if you have a circulatory problem, then you stop using oxygen and you have to burn a lot more glucose to get the same amount of ATP. Right, so that 0.3 millimolar is a sort of underestimate when there's an availability problem, right? And then the other reason that all of this is kind of an underestimate is this is averaged over the whole brain. And at any one time, there's this old idea that, oh, you never use more than 10% of your brain. That's a crock, right? But at any one time, your whole brain isn't super active. I mean, maybe the chess players manage to activate a higher fraction of their brain at once, and that's why they lose weight during a tournament. But if you look at functional MRI, and we'll look at that in a few minutes, it's basically just a piece of the brain, parts of the brain that get activated at once, and so this very high energy usage is averaged over the whole brain, some of which is active, some of which is not. So if you think about the intensity of energy usage in a small part of the brain, it's even more intense than this. So the energy demand of each brain region and each neuron is highly dynamic, right? When neurons get activated, they use a lot more energy. Okay, so that's basically the overall constraints on the system. I think it's really cool to think of it. I mean, as I said, in evolutionary terms, this is a big investment, right? The brain, having a brain that burns this much energy is a big investment, and it must have a good payoff. And we know kind of what the payoffs are. I mean, we're pretty good at getting away from predators and others, and finding food, and outsmarting other things and bad situations. And so it's worth it. But it's also interesting from an engineering standpoint, right? That biology had to do a lot of good engineering, both in terms of efficiency and flexibility and regulation, in order to make this all work. So now, what does this look like? We're gonna go down. This is a very vague general thing. What does this look like at the cellular level? And we're gonna start out with energy demand. So what is so demanding about running a brain? And I'm just gonna remind you of, this is what I teach. This is what I usually teach to first-year graduate students. I'm going to remind you about electrical signaling and how electrical signaling works. Electrical signaling is the thing that makes our nervous systems remarkable, right? It lets us coordinate movements over large distances with, in a fraction of a second. And that's only electrical signaling could do that. If I made some cyclic AMP in a central neuron, then for that cyclic AMP, it would take hours to get out to the terminal processes, but instead it takes milliseconds or hundreds of milliseconds to be propagated in the nerve. So how does fast electrical signaling work? And it takes two ingredients. And one ingredient is ion gradients, and the other is selective permeability. And so ion gradients are the big difference in ion concentrations inside and outside cells. Sodium is much higher on the outside, potassium is much higher on the inside of cells. And that actually has to be set up by ion pumps, doing sort of manual, one bucket at a time, bailing of sodium from the cell and taking potassium in. And each 3 sodium ions that gets pumped out requires 1 ATP. And these pumps, there are lots of these pumps in the plasma membrane. The cartoon is of the sodium-potassium ATPase, or the sodium pump. And it is probably the most important pump, but there are a few other primary ATPases, and it pumps uphill, so it needs energy. You can't go against randomness without investing some energy. And so these work, just to set up the situation, just to get my ion gradients where they are now, required a big initial investment. Maintaining them requires an ongoing investment. And it's slow, but slow doesn't mean much, because that's slow on a per molecule basis. So each molecule of sodium pump of even each protein can only move a couple hundred ions per second, a couple hundred turnovers per second. What is the selective permeability part is how we get really fast electrical signaling, and it's done by ion channels. And the ion channels are highly selective to which ion goes through them. So the cartoon here is of a sodium channel, and a sodium channel lets only sodium go through, and it only goes down its electrochemical gradient. It's completely passive. But the trade-off is it's completely passive, but it's super, super fast. So one ion channel can move, basically a picoamp is a typical current through an ion channel. And that corresponds to a single ion channel, single protein. That corresponds to 6,000 ions per millisecond, or 6 million ions per second. So you can undo the work of one pump pretty quickly, right? When you open one ion channel. So you need lots of pumps there. And it says that this is very energy demanding. Another way to use this calculation is this is 2 million, one ion channel can be responsible for you having to burn 2 million ATPs, right, in a second. 2 million ATPs. So it's a lot of ATP. This is the main expense of running a nervous system. It's basically a battery, right? It's just like when I recharge the lithium battery in my MacBook. This is the battery charging, right? You're pumping ions, you're separating ions. It's actually exactly what happens in a battery. You're separating ions so that you can use that voltage to do useful work through the passive mechanisms, in this case, through transistors on the chips. Okay, what does this look like in terms of energy usage? How do we now look at a cell and figure out, or cells in the brain, neurons in the brain, and figure out what their energy usage is? Most of the brain energy, the calculations say, are used to restore ion gradients. So David Atwell's lab has been in the forefront over many years in doing this kind of calculation. I want to warn you that you should take all these numbers with a grain of salt. They're sort of like, if they're very fancy back of the envelope calculations. You say, well, if you've got 2 million of these and 5 million of these, and on the average you should be doing this. But it's still kind of impressive. And the reason you should take it with a grain of salt is that the numbers on this pie chart have changed several times and probably will change several times more over years. So, but what you can see is that this is the ATP required to restore the ion fluxes that flow downhill in a synaptic terminal. So through glutamate activated channels, sodium and calcium enter the cell. The sodium has to be pumped out by the sodium pump. The calcium can become pumped out by calcium ATPases or by sodium calcium exchange, and then through the sodium pump. And so that's that part of the calculation. That says that's half the energy of running your brain is restoring those ions. When they get out of place because of synaptic activity, this is pumping them back into place. This is when you have action potentials, and they were using a calculation like, well, on average, a neuron in the brain fires 4 times a second. And so that's the number. That number changed a huge amount, partly because of work by Bruce Bean and his colleagues here, because they figured out that action potentials in mammalian neurons are actually much more efficient than action potentials in squid axons. There's a lot less overlap between sodium and potassium currents, and so you don't run the battery down as much. So that number changed a lot. But anyway, so this is sodium and action potentials. Sodium that just maintaining the resting potential, there's like a little bit of leakage in the wrong direction. You have to keep restoring that. Presynaptic calcium, when calcium comes into the, sorry, this is from the Atwell paper. I tried to keep it simple. This is the actual figure from Atwell. And calcium that enters the presynaptic terminal, super important for signaling, has to get pumped back out again. And they calculated that as 4%. So this is 96% of this is pumping ions that flow in across the plasma membrane, pumping them back out. They attributed 4% to neurotransmitter recycling, which is also a pumping process, right? There's a sodium-dependent pumping process there. There's vesicular proton ATPases, and probably this number is an underestimate, according to, at least in cultured neurons. It looks like these are leakier than people thought. And so this number is probably bigger. But the overall idea is about right. I mean, there's other stuff that requires energy in neurons, actin, myosin things. The actual vesicle cycling is energy requiring. But the numbers of ATP required for those processes are probably quite small compared to these big ion fluxes. Okay, so that's what demand looks like in the system. What about energy supply? So where do we get all that ATP? And the answer is glucose, most of the time, is the main fuel for the nervous system, for the brain. And this is like a whole biochemistry course, and some of you are probably having PTSD from just remembering your sophomore biochemistry course, but I'm going to break it down into just a few modules and not show you all the steps. So glucose goes through a few steps in the cytosol called glycolysis and gets converted to 2 molecules of pyruvate. And glucose, this glycolysis, this process of glycolysis, you have to invest 2 ATP and you get 4 ATP back. That's right. So it doesn't give you a lot of net ATP, but it does give you some. And as we'll discuss, it can do it pretty fast. So this is part 1. It happens in the cytosol called glycolysis, doesn't require oxygen. Part 2, sorry, not part 2 yet. So if you don't have the rest, if you're just doing glycolysis, you need a way of recycling the NAD+. NAD+ is a cofactor that moves electrons around the cell. And you need a way of recycling it where this will stop going forward. And what you can do is just recycle that NAD+. Turns out that NADH is really valuable. Each NADH could give you like 2.5 ATP molecules. And you get 2 molecules of NADH from glycolysis. But you can't do that if you don't have mitochondria yet. So you have to recycle, or you don't have oxygen. You have to recycle the NADH by converting pyruvate into lactate. Okay, here's the big dog for making ATP is the mitochondria in the cell. And pyruvate gets taken up by the mitochondria, goes through a set of biochemical reactions, the Krebs cycle, tricarboxylic acid cycle. That doesn't make much ATP. You actually get 2 GTP molecules out of that. You can cash them in for ATP, but that's not where most of the energy comes from. Again, most of the energy comes from electrons that are transferred to NADH, and you don't get ATP directly from that. What you get from that is proton pumping, right? So this is the Mitchell-Chemiosmotic hypothesis. Protons get pumped out of the mitochondria, alkalinizes the inside of the mitochondria. You get 10 protons for each NADH. And then you've got this amazing motor called the ATP synthase. And it's like a water wheel where protons flowing back in through the membrane, these protons that got pumped out flow back in through the membrane. They actually turn a rotary motor, which drives ATP synthesis. So ATP gets synthesized, it has to get out of the mitochondria to do the work that we need it to do. But that's sort of the general scheme. Total numbers, when this system is running, when you don't have to do this, the system is running completely, you get 2 ATP from glycolysis directly. Some of these NADHs actually get shuttled to the mitochondria. It costs you a little bit because you need a proton to pump this whole process, but you get more, so you get more ATP out of that. You only get 2 directly. You get another 29 from this process. Okay, so this is where most of the ATP comes from stoichiometrically. Okay, that wasn't so bad, right? That's the basic process. Now, there's some flexibility in how these pathways are used, even when your only fuel is glucose. So on average, when things are kind of at rest in the brain, you burn glucose completely. I didn't draw it, but you get a couple of molecules of carbon dioxide out here. So this process of electron transport transfers these electrons to oxygen, to molecular oxygen. And so you consume oxygen here and you produce CO2 here. And so the end result is you turn glucose entirely into carbon dioxide. And when you do that, when you use this whole pathway, that's about what the brain does at rest. It's not perfect. So there's still, the stoichiometry isn't perfect, but it's pretty close to that being, glucose being burned 100%. But if you don't have oxygen, or it turns out if you're in a rush, then you could do this. You could burn glucose to lactate. And we'll talk a little bit more about that in a few minutes. So you could burn glucose to lactate. Well, then what happens to the lactate? Well, lactate is actually too valuable to waste, right? You've only gotten 2 of the 31 ATP that you can get out of glucose if you stop at lactate. And what almost certainly happens to lactate, the brain doesn't excrete much lactate under normal circumstances. And so even if you're in a rush and lactate is temporarily being produced, that lactate just goes back in, right? That lactate probably goes back in and goes the rest of the way. And whether it does it in the same cell or it does it in another cell is a huge source of debate, which we'll come to in 2 sections of the talk. But this gives you some flexibility, because to use the purple line, you have to have a perfect match in the rate of glycolysis and the rate of oxidative phosphorylation. This whole process is called oxidative phosphorylation. And if you want to run this a little bit faster, then something's got to give, and you temporarily produce lactate, but then it will be used because it's not really a waste product. Okay, last bit of cellular energy production are all the exceptions to the rule that glucose is the main fuel. And so there are other fuels available. I already told you about glycogen. Glycogen enters here. So glycogen, it skips one investment step, so it's a little cheaper to use. You get a little more net ATP out of it. But as I said, you can't run on glycogen for long. It's not even clear, there's a lot of questions about glycogen. Let's just leave it at that. Ketone bodies, which we'll talk about, the ketogenic diet, or fatty acids, enter metabolism at the acetyl-CoA step. So they skip glycolysis and they enter there. We'll talk about that in a few minutes. Lactate, so lactate in principle is used as a fuel by the brain, but the brain actually takes very little lactate up from the circulation, even though there is lactate in the circulation. Very nice paper by Josh Rabinowitz and his colleagues showing that lactate is really shared among peripheral organs, right? That there are a lot of peripheral organs, just burn glucose to lactate, send the lactate out in the blood, somebody else uses the lactate. Sometimes they use the lactate, but it's shared. The brain doesn't do that. The brain pretty much burns its glucose, and any of the lactate that's generated is probably used locally. So that's kind of interesting. But lactate, will come back to the idea of lactate as a fuel, and you know what it'll do, right? It'll go through glycolysis. And then things that people don't really think about as being fuels, glutamate and glutamine, I mean, they're amino acids, and they also tie directly in to the tricarboxylic acid cycle, to the Krebs cycle, and they can be burned as fuels. And they're in abundant supply, right? Glutamate, so it's a neurotransmitter. You don't want to put you don't want to feed the brain glutamate directly because you'll get it all excited. You can feed the brain glutamine, and that's how glutamate comes back from glia. Right, if glia take up the glutamine, and then they send it back. They sort of ship it back to the neurons in a safe form, right? And these can be used as fuels. You can't do that for a long time because you have to maintain your glutamate pool. But that can be done by using glucose, and instead of oxidizing it all the way, you produce glutamate through the same set of pathways. Okay, so that's basically the end of the bottom up section. This is you now have some understanding of how energy is demanded, right? What is creating the energy demand? Mostly it's the ion pumping, and this is the way you make the ATP. And that doesn't mean we understand everything. In fact, all of the pathways I've shown you are pathways. It's basically what can happen, right? And a lot of this was worked out in the 1950s and 60s. And that's why even people my age figured, oh, this is a done deal. Everybody, we know everything about metabolism. We can ignore the housekeeping stuff and move on. What we still are pretty ignorant of is a lot of the dynamics of the system. right? The actual ebb and flow, not just what can happen, but what does happen. And that's what those of us who work in the area are trying to figure out what actually does happen. Okay, so first thing about what does happen. What happens to energy metabolism when you think hard, or when part of your brain thinks hard? And we're going to start with, again, the top-down analysis of that. So looking at the brain as a whole. So this is functional MRI. You guys have seen lots of these, lots of these images. That's the back part of the brain, the occipital cortex. That's your visual cortex. And if you look at a very disturbing flashing checkerboard display, it really is very unpleasant to look at. But if you're looking at it and you're sitting in a scanner, your brain will do that. You'll get a big delta, a big increase in the bold signal, in the blood oxygenation level signal, which is used for all functional MRI in the back of your brain, representing increased activity in that part of the brain. Now, what's funny about that bold signal, so the bold signal, you can see a magnetic resonance signal from blood oxygenation because deoxyglucose is paramagnetic. Deoxyhemoglobin. Oxygenated hemoglobin is not paramagnetic, so you get a signal. But what's weird about that bold signal is that you have more oxygen in the area with increased metabolism, which is weird, right? Because that area should be using more oxygen. It has higher metabolism if you think about it in a simple way. And there's a resolution to this puzzle. It's kind of surprising, right? It's kind of surprising that that's not a direct reflection of increased metabolism, but there's another top-down tool for looking at this called positron emission tomography, where you use radio tracers to monitor a lot of things, right? You can monitor blood flow by using a tracer that basically just remains in the blood. And what you can see in that same kind of experiment is a classic paper by Fox, Rakeley, Minton, and Dentz that shows that in this same kind of experiment, blood flow to the back of your brain increased by 50%, okay? And glucose uptake increased by 50%. So far, so good. All corresponds to increased metabolism. But surprisingly, and they had to sort of tune up their scale because there wasn't a lot of oxygen change, oxygen uptake in that part of the brain only increased by 5%. Okay, so that means their metabolism is not working the same way when this part of the brain gets stimulated. Makes sense for the bold signal because you're delivering more oxygen, but you're not removing as much oxygen. So that's why the blood is more oxygenated in the active part of the brain. Okay, what does this mean in terms of the microscopic, in terms of the cellular level stuff? Well, I told you that at rest, the brain is burning a pretty good match of oxygen and glucose, right? So that the glucose is being completely metabolized. But now we learned that when you stimulate locally, glucose consumption goes up by 50%, but oxygen consumption only goes up by 5%. This is exactly the thing I have prepared you to think about, which is what happens when you have a mismatch between the rate of glucose consumption and the rate of oxidative phosphorylation. And the answer is something's got to give. And in order to keep glycolysis going at this elevated rate, you need to recycle your NAD+ by producing lactate. So temporarily, and this actually happens, right? Temporarily, when in the active part of the brain, you're doing what's called aerobic glycolysis. It's glycolysis. It goes from glucose to lactate, but it's happening even though there's plenty of oxygen around. The brain is making a decision to do that because it's running glycolysis faster than it can take away pyruvate and NADH with the mitochondria. So this kind of metabolism, this kind of aerobic glycolysis was described in cancer in the 1940s, right? That some kinds of cancer cells, not all cancer cells, do this. They do more glycolysis than they do oxidative phosphorylation. So that's it's a little surprising. I told you, you're only getting 2 ATP per glucose when you do this. So it's a little surprising. We'll come back to that. But right now I'm going to switch gears and ask the question, so at the cellular level, what's actually going on when this happens? So we're going to switch to how might multiple cell types be involved? And there was a pretty popular theory that well, here's the simple idea is this is all going on in neurons, right? That's basically how I presented you one cell, right? And that one typical cell is doing this, and when it gets stimulated, it does it more. Sorry, I should have done that. So neuronal glycolysis, going back to lactate. This other hypothesis, been around about 30 years, exactly 30 years. It's called the astrocyte neuron lactate shuttle. And the idea is, now this isn't all happening in one cell. The glycolysis part is happening in the astrocytes, and then lactate metabolism is happening in neurons. And so the idea is, when you stimulate neurons synaptically with glutamate, that glutamate gets taken up by astrocytes. That stimulates the astrocytes to do glycolysis, to convert glucose to lactate. And the idea of the shuttle is that that's the main source of food for an activated neuron, right? That lactate is delivered, it's sort of chewed up by the astrocyte and delivered to the baby birds, right? Delivered to the neurons to use as fuel. And so that's the astrocyte neuron lactate shuttle. This obviously is, if it's right, it's very important to understand, right? It means lactate is moving between cells. And we asked the question, so I'm going to tell you a little bit of our research now about which of these is going on. Both of these hypotheses predict an increase in cytosolic NADH, and we can measure that using fluorescent biosensors. So to get this part, to get the underlying information in this part of the talk, you need to take Biosensors 101. I don't know when that's going to be offered, but in Biosensors 101, we taught you how to make fluorescent biosensors for different things, but I'm just going to show you the results. So this is in a hippocampal slice. We have two kinds of biosensors in the slice, one that detects calcium and the other that detects NADH. And so the prediction from either of those models is that after we stimulate a neuron, we should see a big increase in the cytosolic NADH. And here you go. So there's just a quick flash on these two neurons. There's a quick flash and we excite them. And then watch these two neurons, and then there's a bloom of NADH that lasts for several minutes. And so it's being plotted up there, right? This is the flash of calcium and the several minutes elevation of NADH. So we are seeing this change, and we see it very reliably. This is the average behavior. It depends very much, the increase in calcium and the increase in NADH are very highly correlated with each other, right? So now the question is, which of those models is the right model? And I'm just going to summarize with what I consider the most critical experiment. So we want to distinguish between this happening, the neuron burning its own glucose, and transfer of lactate from one cell to the other. So first of all, can the neuron respond? So our sensor's in the neuron. Can the neuron respond when we deliver extracellular lactate? The answer is yes. We can get a big increase in NADH, comparable to what we get with stimulation, a big increase in cytosolic NADH when we stimulate. And now the critical experiment is to block the lactate transfer from one cell to another. And the prediction is that we should get rid of, if this was the astrocyte neuron shuttle was right, we should get rid of this. And first we check to make sure this blocker is blocking the response to extracellular, just exogenous lactate, and it does. And then we do the critical experiment and we stimulate, and no, it didn't go away, right? This did not go away when we blocked that transporter. So it's almost certainly this. And in fact, the change got bigger, and that's probably because what's going on is the neuron is doing this, and then we're bottling up the lactate, which puts back pressure on this reaction. So the summary of this part is The astrocyte neuron lactate shuttle is not the source of the transients. Neurons do their own glycolysis, and probably at least during excitation, they export it, although then they and other cells can take it back up. So for the last two parts, the question is, what's going on? Why would neurons do this very inefficient thing of only using glycolysis? It only gives them 2 ATP per glucose instead of going the whole way. And I already suggested the answer, which is it might be faster. And it's a lot like the neuron needs to sprint. When you sprint, let's say when you take off from a red light in your car, if you're monitoring your energy efficiency is terrible, but it lets you go fast. That's probably what the neuron is doing. And the analogy to a sprint, to running sprint, is actually not accidental. If glycolysis is really the fast energy production, then muscle does this too. So flight muscle in birds is the white meat of the bird, right? The turkey. And it's white because it has very few mitochondria compared to the postural muscle, which is red. So muscle that needs to go really fast to make energy really fast depends much more on glycolysis. As I said, the lactate is not a waste product. It probably gets used by everybody afterwards. And I'm just going to skip ahead there. Okay, so I've left myself very little time, like no time, because Perazad wants me to leave time for questions, and we have a question right here.
I just have a quick question. So how do the neurons buffer the proton when they're making that lactic acid really fast?
So with buffers.
Okay.
I mean, there's no other answer. They do acidify a little bit when you stimulate them. Okay. And the protons get exported too. So the transporter for lactate also transports a proton. Right, so you get extracellular acidification. It matters. It's a huge problem for biosensors. Is making sure that pH change doesn't interfere, but it's a manageable pH change. Great question, though. Okay, I'm gonna just really quickly suggest the answer so we can get to questions. As Perazad mentioned, even in drug-resistant epilepsy is super common. Even in drug-resistant epilepsy, going on a ketogenic diet can be really valuable for seizure control. And the reason not everybody does this is it's actually a very strict diet. Nobody does strict diets, so nobody wants to do this. That's a ketogenic diet. It's a few little crackers per day for a 6-year-old. What happens on this diet is your liver produces these 4-chain so-called ketone bodies, these 2 molecules at millimolar levels. They circulate at millimolar levels in your blood at fuel source levels. And it was established years ago, they could be used in lieu of glucose to produce more than half of your brain's energy. So that's a big exception to the rule that I told you about glucose being the main fuel. I'm going to really summarize here is that we think that this change in fuel underlies the therapeutic effect. And so that's your brain on glucose. You already know that ketone bodies do that directly. What I didn't mention is that whenever you rev up the mitochondria, you inhibit glycolysis. You inhibit glucose utilization. So how might that help neurons? Well, it might help neurons from firing fast. I'm skipping a bunch of stuff by opening ATP-sensitive potassium channels. So these are channels that are closed by ATP. And they're really well known in pancreatic beta cells where they control insulin release. They're open between meals and keep the cells non-excited, but when you have a sugary meal, they get closed, and in those cells, that's enough to just make them fire action potential. In the brain, these channels are found in many neurons, but in well-fed brain, it's a puzzle, right? Everybody says, oh, no, there's enough ATP to keep these channels closed. So how might the fuel switch help? Well, the answer is there might be a division of labor between ATP produced by glycolysis in ATP produced by oxidative phosphorylation. And in fact, glycolytic enzymes can be found very close to these channels. And so when you have ketone bodies present and you diminish the amount of glycolysis, that can reduce local ATP and open those channels. And it's kind of cool that that local ATP will also be influenced if you're starting to have a seizure, you have burning a lot more ATP near the plasma membrane, and that can also lower ATP. So those two things together can open these channels, and because they're potassium channels, they reduce excitability. And just very quick summary, because I'm out of time, that's what we see. In a model, in a metabolic mouse model that mimics the ketogenic diet, we see ATP channels are much more open. We see that individual neurons' excitability is dramatically affected, and that difference goes away if you block ATP channels, so it's entirely dependent on that. We see seizure-like activity in slices that, again, is dependent on this metabolic change and goes away if you block ATP channels, and the seizure resistance in the whole mouse goes away if you block if you get rid of ATP channels by genetically knocking them out. Okay, I am done with the main subject matter, but I have I'm gonna use 2 more minutes. I want to make the point that courses taught by professors tend to emphasize how much we know. And that is complementary to us professors, because we've made some contribution to understanding what we know. But I think it's really important to emphasize how much we don't know. And every graduate student who listens to a teaching course gets demoralized because it sounds like all the good stuff is done. But in fact, we have a lot of questions left. So I'm just going to show you 5 things that are challenges. And it's not that we have no answers, but we have incomplete answers. One of them is what pathways get used and when. And I've been talking about that, but it's actually really challenging to measure, because even the biosensors, which are really good, only tell us the levels of things. They don't tell us how much is going through this pathway and how much is going through that pathway. We can use carbon tracing to do that, but it's challenging to do it in intact tissue. 2, regulatory control. What controls these things? Again, we have some hints, but an incomplete picture. 3, in disease, is altered metabolism that's often seen in disease, let's say in neurodegenerative disease. Very common reports, glucose metabolism is impaired in normal aging and worse in Alzheimer's. But is that a consequence of the neurodegeneration, or is it a cause of the neurodegeneration? We don't know, and there's ample reason to suspect that it might partly be a cause. If it is a cause, how do we fix it? Do we bypass it? Like in the ketogenic diet, is the problem energy supply, or do we try to boost glucose metabolism? As I showed you with the ketogenic diet, those have different consequences, and the choice is important. And finally, I haven't mentioned, but many of the same people who did the cancer metabolism stuff realized that epigenetics, acetylation, methylation of histones, let's say, is very sensitive to metabolic state of the cell. And so that's a whole interesting area that I haven't talked about at all. So I'm going to stop there. I'm going to thank, because I did talk very briefly about 2 research things, I'm going to thank Carlos Manlio Diaz Garcia, who is the first author on the biosensor study where we looked at the astrocyte neuron lactate shuttle, and Juan Martinez Francois, who's right there, who did the work on BAD and the ketogenic diet. But I'm gonna stop there and take questions. So thanks. Sorry to go over. Yeah.
So lactate, where the lactate goes?
Where does the lactate go? So as I suggested, I had that pathway slide where I showed lactate being produced and then lactate coming back in and being consumed. I think that's what happens to it. And I think everybody uses lactate. I mean, lactate's good stuff, right? So everybody, if the lactate is extracellular, I think the neuron that just made it will use it. But I think the neighboring astrocytes will use it. I think the other neurons that weren't excited and didn't make lactate will use it.
you think about this oxygen consumption, right, the calculation that we make, it doesn't really, right, lactate cannot be used unless oxygen.
Exactly, but I think it's in the noise, right? So, because I think slow utilization of lactate after the fast production of lactate, I think you don't see that, right? You're right. You'd predict, if that 5% increase in oxygen lasts a little bit longer than the 50% increase in glucose, then that would be enough to chew up the lactate, right? It's got to last longer. I don't know if it does, but it's like very close to the noise.
It's muscle, right? You lactate goes to liver, genesis.
That's right. That's right. So as I said, this Rabinowitz paper says that peripheral organs share lactate, but most of the lactate made in the brain gets used in the brain. The brain excretes very little lactate, even with all this going on. And so that's why I think it's just getting used locally. But there's not good evidence for it. So this is one of the things, people should pay attention and figure out what's happening to it. We'd like to know. It's a great question.
And is there a reason why brain retains lactate? Is because the lactate pod is more widely expressed in neurons or more efficiently imports lactate, or is that because lactate is not as permeable through blood-brain barriers?
So the question is, why does lactate get retained? And I think the permeability is very high everywhere. Right, permeability is high in capillaries, and permeability, they all use the same monocarboxylate or the same mix of monocarboxylate transporters. I think the reason is that lactate is avidly used, right? It's being, it's in equilibrium between, or very close to equilibrium between these places. And when a neuron, or an astrocyte has lactate, it just uses it. So I think it's just the rate of consumption would be the argument rather than anything else. There's no strict, it's not like they have a sodium dependent uptake and the endothelia don't. I think it's just the statistics of who they encounter.
An alternative question is probably you also capture rusty gauges, ion neurons. So they basically spotted this Warburg-like effect in the old neurons. And how come, I think cancer use the sort of this Warburg effect to survive better. How come that is something bad for the neurons?
Well, we don't know is the main answer. But in cancer, Warburg metabolism is almost certainly not about energy. In cancer, it's probably about producing things, right? It's producing intermediates. I didn't show, as I said, I was only talking about energy metabolism, so I didn't show all the goodies that you can get by burning glucose. So glucose, for instance, is very important for making pentoses like ribose that you need to make DNA. It's also important for making amino acids. And so probably a lot of the reason glycolysis is important in developing cells and in cancer is actually the byproducts rather than the ATP itself. Okay, but we basically don't understand. We don't even know if it's bad in the older neurons, or is it just that older neurons don't do it, have other problems, and that's what we need to, that's what we need to figure out. We don't know if it's part of the disease or aging process. Is it just a byproduct of the aging process, or is it part of the reason aging is bad? Yes.
So in the ketogenic diet, could it in the long term cause some negative effects?
So the question is, does ketogenic diet in the long term cause negative effects? And the answer is possibly. So it depends on what kind of fats you're eating. And so, there probably are some, fat accumulation effects due to that. But another part of the answer to that is that very often epilepsy patients don't need to stay on the diet forever. In many kinds of epilepsy, regardless of how you treat the seizures, if you can get rid of the seizures for a couple of years, you can taper whatever it is you've done to get rid of them, and often the seizures don't come back. And it's not always true, but it's sometimes true. The other thing to remark is that there are metabolic syndromes for which the diet is used, and it has to be used forever. And that is, the best example is the GLUT1 deficiency syndrome. So it's a glucose transporter deficiency where glucose doesn't get into the brain very well. And so in that case, you're supplying an alternative fuel and that's needed for brain activity, that need never goes away in those patients. And so they stay on the diet forever. But the diet can be managed. I mean, the diet, I said it was hard to do. It's not just hard to do. It actually has to be managed by dietitians to make sure you're getting the right combinations of things. You need different sets of vitamins to burn that much lipid as fuel. And so it needs to be managed carefully, but it can be fine for an extended time. Yes.
I kind of follow up on the same sort of line of questioning. For ketogenic diet, like how long lasting the effects are of this switch in the metabolism? How much durability there for the change of metabolite for these neurons?
Yeah, that's a great question. The question is how long? I'm sorry.
And also, if the excitability and properties of neuron changes, does it cause like circuit rearrangements in the
Right. So two-part question, right? One is how long are the effects of the ketogenic diet on metabolism? And second, how long lasting are the effects on neuronal circuitry? So the first part of the question is that the ketogenic diet effects on metabolism can be very rapidly reversed. And if a kid on the diet, because it's mostly kids who do the diet, I think, because their parents make them, whereas adults, we're very bad at diets. But if a kid on the diet cheats and has a candy bar, they lose their protection within the hour. And that is the time scale on which circulating ketone bodies disappear. Okay, so, and that was one of the things that made me think, well, maybe it's really an acute physiologic effect of fuel usage rather than thousands of little changes in gene expression that happen, because diet is a very complex manipulation. Your other question was about changes in circuitry, and it goes to my other question, the other question about do you have to stay on the diet forever? And the fact is that seizures, I mean, for whatever reason they're caused, seizures are also a learned behavior. So just like anything else, the brain learns to do seizures really well. Those pathways get reinforced. That's the way we learn stuff. And so the brain is very good at reinforcing pathways. If you can prevent the seizures, that learning disappears. And that's the reason you can remove the treatment. So that's a quick answer to your question, but it's the best one I know.
Thank you.
Yeah. Here's that.
So we have some questions on Zoom. So to be fair, I'll read some of those out.
Okay.
So let's see, ton to choose from. Let's see. So you showed the brain metabolism in a cellular level. Since the brain has receptors for bile acids, my question is if there is an interaction between cholesterol metabolism and brain metabolism.
Wow, I don't know the answer to that question, so I'll answer a different question. The different question I'll answer is that the brain has receptors for lactate, has GPCRs for lactate that are probably important in signaling, but we don't know quite what role they play. I just don't know the answer to the other to the question that was asked. So
Yeah.
I answered a different question. Good pro tip for answering the question.
So the same person also asked the second question, which is about the technique to image the fluorescent probes in the brain.
Yeah.
So I think maybe they mean the biosensors, like how.
Okay, so the biosensors are fluorescence.
Mhm.
So you can use 2-photon microscopy. So the images I showed were in a brain slice, but we've also done in vivo, where you put a window in the skull. You express. So first thing is you have to express the sensors. The sensors are genetically encoded, and they're expressed by injecting a viral vector that injects the DNA into cells, and then the cells make the sensor. Then to image them, we use standard fluorescence imaging, like 2-photon microscopy, that gives you an optical section within the brain slice. So we get it's as though we have an optical section. And then the last comment on that is that all the fluorescent biosensors that we use have a change in what's called fluorescence lifetime, because it's not enough to look. If you look at the brightness changing of a sensor, the brightness can change, or let's say one cell can be brighter than another because it has more NADH. but it can also be brighter than another because it has more sensor protein. And so we have different ways of correcting for that, but our favorite is to use fluorescence lifetime, which changes independent of the amount of sensor that's present. So those are the quick answers to that series of questions.
Thank you.
Sure. We have one more here. Somebody, who did I miss? Yes.
I have two questions related for the relationship between the metabolism and cell excitability. So is there any way to pick up the basal, like if there is an increased basal activity of the neurons, can we pick up this? Like what exactly influences basal, like?
Yes, so it's a great question. You know, I think there are bigger differences in excited cell firing than there are in basal cell firing.
Yes.
But when we started working on this, we looked at spontaneously active neurons that have pretty high basal cell firing, and their basal cell firing is also affected. Okay, but it's probably also because when you have a high basal firing rate, you have more ATP usage right at the cell membrane. And I should emphasize that's still like our working hypothesis that it's local ATP. I don't feel we have a complete understanding of what's going on there, but that hypothesis, you know, we've been testing different versions of that hypothesis for many years now, and it's still not wrong. It's not right, but it's still not wrong. I mean, it's not proven, but it's still not wrong. And we'd like more direct evidence for what's going on. But yeah, that's the basic answer to your question.
Another question
Yep.
is, within the neuron, is there any heterogeneity, like limited to the soma region, or like?
That's a great question. So the question is this the same in different parts of the neuron? And we have looked at the lactate shuttle question in different parts of the neuron. In the same neurons, we've looked at the somata, the dendrites, and the axon terminals. And in all three cases, there are subtle differences between them, but in all three cases, we see that the NADH, there's an NADH transient, and the NADH transient does not go away, doesn't get smaller when we block monocarboxylate transport. Usually it gets bigger. Okay, so in the neurons we've studied, in the parts of the neurons, we haven't seen a difference between the different parts of the neurons that we've looked.
Oh, thank you.
Okay, yeah.
Is there a genetic basis or reason why the ketogenic diet works for some but not others, in a sense? Is it in the ability to block glycolysis, or is that correlated?
So nobody knows. So the question is there a genetic basis for why the diet works in some people and not in others? And we were really interested in this. For instance, there is genetic heterogeneity in the KATP channels. And we did a very small study, like 60 patients, at trying to see if there was a correlation between that common heterogeneity in KATP channels and success of the diet, and we didn't see anything. But it's very underpowered, so I don't rule out that. I think that could be one reason that it's true, but we don't know the answer. It's the short answer.
You showed those early data, still after stimulation, brain consume more glucose, but only a little bit oxygen. I wonder if that is a general phenomenon. Think about muscle. If we do exercise, is that also happening?
Oh yeah, so that's the other analogy to sprint, is that if you sprint, you can't sprint forever, right? Usain Bolt can't run a marathon. Maybe he probably can run a marathon, but he can't run it as fast as he can run 100 meters. And so you can't sustain that level of activity, and you produce a lot of lactate. You get cramps, right? You get lactate because you're doing more glycolysis than you are oxidative metabolism, so you don't keep up. So muscle, absolutely. I mean, and it's not just a difference in time, it's also a difference in fast twitch muscle versus slow twitch muscle, right? And postural muscle versus flight muscle is the most obvious example in birds.
So I guess, I was thinking maybe, this neuron might have a different requirement for the oxygen versus glucose.
Mhm.
I wonder if that, it is the same thing for other energy consuming
Organ.
cell types,
So
like muscle or the beta cell.
right, so muscle, as they say, muscle, there are subtypes of muscle that are specialized for either, burning as much, using as much oxygen as glucose or using glucose in preference. So that's the white muscle versus the red muscle. So that's more obvious than neurons. I think that probably all neurons use, do complete glucose oxidation at rest. Mitochondria there's a lot of mitochondria. There are mitochondria at maybe half of central synapses, not all central synapses. If you ask Tom Schwartz, he'll probably, I think the best studies say that at any one time, only half your synapses, presynaptic terminals will have a mitochondria in there. But I think the neurons are more facile in their switching between these conditions, whereas muscles can be specialists. So I hope that answers your question. Yeah.
Unfortunately, I think we have to end, but there's a ton of questions on Zoom as well. It's a really exciting topic. I'll save the chat for you so you can
Okay,
see some of
perfect.
them.
I'm
Perhaps
happy.
people can.
Yeah, if people want to send me email or something like that, I'm happy to respond.
Yeah, thank you so much.
Okay, great.
No.